So hello everyone. Hopefully with this presentation today we're going to tie up a few things together. So in the previous chapter we talked about how DNA is replicated and also how DNA is transformed into RNA and then into proteins. So what we're going to do today is mostly look at all of this, but in a context of gene control, gene expression, but also how loss of this control and how this control can be lost can lead to, of course, cancer, unfortunately. Cancer is something we're all in some way or another familiar with. Most of us have someone in our lives that has been struggling with cancer, or maybe is struggling right now, or maybe people passed away because of cancer. So the goal today is not to get so much in the cancer biology, but to explain how cancer starts and what's at the origin of cancer. There are many different causes of cancer, and of course we're not going to get into all of that. You know, we could spend hours and hours and hours and lecture after lecture talking about cancer. It can be very advanced. It's a very it's a side of science that's very, very active, obviously, because so many people are struggling with cancer. So, but today is will be mostly about how to explain the basics of mutations. What are mutations? What kind of mutations we can find? But also how mutations can affect gene expression, and of course that can lead to cancer. So, let's start with the next part. So we have seen that there is something called the central dogma of biology. And the central dogma of biology goes like this. So we have a DNA molecule, and that DNA molecule can be replicated. So basically, when we go from one cell and then we divide the cell into two cells, right? So that's a process of mitosis. So when we go from one cell to two cells through mitosis, both of those daughter cells here need to have the same amount of DNA. So basically, the most important thing here would be to replicate this DNA to make sure that every single of those daughter cells, this one and this one, have the same amount of DNA as these ones here. So that is the process of replication, and there is a very definite process of this. We've seen that DNA polymerase 3 is involved, and we've seen also that replication of DNA is a process that's not continuous. What it means is that when we have a DNA that's double-stranded, one of the strands is synthesized continuously. The enzyme starts at one end and then moves to the other end, but the other strand of DNA, because of the orientation of it, needs to be synthesized in an interrupted way, and it forms the Okazaki fragments, as we have seen. Okay, so that is important, that if you're not familiar with this, or if you struggle with this and you think, wow, I'm not sure about that, I think it's important to stop the video right now and basically go back to that and make sure that you understand that. Now, once the cells have their own DNA, the goal of DNA really is to keep up with the instructions of the cells, right? So whatever a cell is, so let's say we have a liver cell. Well, the liver cell knows it's a liver cell because genes expressed in that cell tells the cell that it is a liver cell. It has certain proteins that make this cell a liver cell. Same thing with brain cells. Now, DNA in itself doesn't have much of a function. The function of any cell and everything that we see in cells is basically given by proteins. Proteins are the workers of the cell. This is where the action happens. The proteins are actually making all those changes. But in order to have a protein from DNA, we need to have some kind of intermediate in the middle here. And that intermediate will be RNA. And more specifically here, it will be a messenger RNA. And the messenger RNA is synthesized from DNA using an enzyme that's called RNA polymerase. And RNA polymerase will interact with DNA at a very specific spot, and that spot is called a promoter. And then the RNA will be synthesized from that one strand of DNA, and that one strand of DNA is called a template strand. And again, if you're not familiar with this, or if you're struggling with this concept, I'm urging you to stop the video right now and then go back to this basic principle. Now, once we have the messenger RNA can be translated. This is a process of translation, can be translated into a protein. So the mRNA here will be read by particles that are called ribosomes. Ribosomes kind of look like this. And then we will have the mRNA here, and the ribosome will be moving on that mRNA and transform this mRNA into a protein. So that protein, so this is an arrow here, and so that protein here will emerge out of that ribosome like that. And then at the end, there's a start where the ribosome starts, and then there's an end here where it stops. And then all of that sequence here will give us a protein like that. So as I talked about the central dogma of molecular biology, which leads from a DNA, which leads to a DNA to an RNA and then a protein, I talked about how the ribosome reads the mRNA. And don't forget that the RNA is the image of one of the strands of DNA. Okay, so that's an important part of that. What we have here on this slide is a piece of RNA. So we have our mRNA right here. So all this long strand here is our RNA. And this is a process of translation. So basically this process of translation is from RNA, mRNA, that will lead to the formation of a protein. And don't forget that this protein has a function. Okay, so it has a job to accomplish inside a cell. No cell will produce a protein that is not useful to it. So what we have here is a particle that's called a ribosome, and you can see that there are two little subunits of the ribosome. So one is the large subunit, the other one is the smaller, or small subunit we can see here. And what happens is that a ribosome, as it flies really on the mRNA, that's very fast. One thing you need to know is that the ribosome really doesn't lose time on that piece of RNA. It really flies through. And what a ribosome does is that it reads the bases, the nucleotides that are found in the mRNA, and then we have these nucleotides right here. Every single little box that you see here is a nucleotide. And what the ribosome does is that it reads those nucleotides 3 by 3. And that three nucleotides is called a codon. Okay, so codon for coding. What it means then is that there's a code there, and that code will lead to the formation of a protein. The ribosome reads that code and understands the code and knows that for every 3 nucleotides, it needs to add a very specific amino acid. And we will see this in just a few slides. So in order to grow the polypeptide chain, and don't forget, polypeptide chain is nothing else but a synonym for a protein. And a polypeptide chain grows amino acid by amino acid. So we have 1, 2, 3, 4, 5, 6, etc., etc. And don't forget that every single one of these amino acids are connected by to one another by something that we call a peptide bond. So what we see here is basically the protein emerging out of the ribosome. And in order to be able to link the amino acids together, we have some kind of an adapter molecule here that's called a transfer RNA. And the transfer RNA can do this. Transfer RNA is a very tiny piece of RNA, And that tiny piece of RNA at the end of it has something that's called the anticodon. So what it does is that it recognizes the codon because based on the complementarity of bases. Remember that whenever you have a G, a guanine, in a DNA or RNA, then it's always facing a cytosine, right? Cytosine. And then if you have an adenine in DNA, then of course it's faced by a thymine. But we know that in RNA, that's important, that in RNA, this adenine is faced by uracil. So if we have, say, a codon that is C, 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 then the anticodon of this would be G, G, and G, right? So this is a complementary sequence of that. And we see this here. So this tRNA fits in a groove inside ribosome here and interacts, its anticodon interacts with the DNA right there, right? With the RNA right there. So we see this right here. There's an interaction right there that happens between the anticodon of the tRNA and the codon found on the mRNA. As the ribosome goes codon by codon, so what it does is it goes to a codon, the tRNA comes in here, interacts with it and adds an amino acid, and then the ribosome moves another codon after, right? 3 more nucleotide next to it, and this goes on and on and on. So basically always 3 by 3. It stops at every 3 nucleotides. So as we go along the mRNA, so this is our mRNA right here. So mRNA right here, right? So we have our mRNA right here. And as we go on the mRNA in this direction right here, don't forget the ribosome would read. So this is my ribosome here, and the ribosome here, the bigger part of the ribosome being right here, right, and the smaller subunit of ribosome being right here. Okay, so we have a ribosome here, and a ribosome is going through this mRNA here. So what it does is that it reads codon by codon, as you can tell here. So it will go from codon number 1, and codon number 1 has this sequence right here. So it goes here, reads those 3 nucleotides, and will add a specific amino acid, which in this case here will be alanine. Once this is done, so when the tRNA interacts with the ribosome, adds the amino acid here, don't forget these are the amino acids right here. So once the amino acid is added to the growing polypeptide chain, then and only then the ribosome will now move on and then stop at the next codon right here. The next codon in this case here is ACG, and ACG codes for another amino acid, and in this case is threonine. And this will go on and on and on, all the way 3 by 3, codon by codon, until the end where it meets this kind of codon here, and this kind of codon is known to be a stop codon. So what it means is that after this codon right here, everything stops. This is the last bus stop. The ribosome goes on and on and on, and sometimes it's like for hundreds and hundreds and hundreds of codons. We have a small one here, but that at a certain very specific spot, the ribosome will stop at this codon right here, and that will be the last stop. This will be added. The codon here the last amino acid to be added will be serine right here, and then no more amino acids are going to be added after, and this will be our polypeptide here. Okay, so our polypeptide, we have this sequence here with all of these amino acids here, but of course the last codon is not coding for any any amino acid, what it tells ribosome is that it's time to move on to another mRNA. So the ribosome, at that point, what's going to happen, the ribosome is going to disassemble. So the big subunit is going to separate from the smaller subunit, and these two particles are now going to move on to another mRNA. So what we're going to look at right now is how to read these codons and know how these codons that we see right here, how these codons translate into the amino acid that we have here. So we can do this by using something that is known as a genetic code. Okay, so this genetic code here can be read with the help of this table here. So now, let's look at the first codon right here. So what we have here is G, and then we have C, and then U. Right? Remember, we're in mRNA, so RNA, instead of having a T, has a uracil right here. So what we're going to do is look at this codon, and we have the first letter here, the second letter here, and of course what we have here is the third letter. So you can look at this table here, and we see that this table is organized in such a way that it has this. It has the first letter here, the second letter here, and then the third letter of each codon right here. Okay, so let's find out how we got to alanine right here. So the first letter, of course, is G. So let's look at G. So what we're going to do is we're going to look at the first letter, and then the first letter here is G. So what we have here is all the codons in this part here, all the codons here that start with G. Okay, so you can tell here every single codons here start with G. Now we're going to look at the second letter of this codon here, and we're going to look at C. Okay, so let's look at that. So let's look at this. We have the second letter, and then all the second letters that starts with C are here, right? So the second letter not that start with C, but the second letter that is C are found here. So what we have here is all of this here, and in this box we have all the codons that have a second letter of C. And then the third letter, of course, that now it's how easy, what the third letter is in this case here, in the case we're interested here, is U. So now we're going to look at this part here, the third letter, and then we see that this third letter is right here. So we have GCU here. Let me choose a right color here. We have GCU right here, and that GCU codes for alanine. GCU alanine. Now we confirm that what we have here is GCU alanine. And then you can move from codon to codon and identify every single one of them. So let's try another one. So what we have here is AGC. The codon number 6, we have AGC. The first letter would be A right here, and then the second letter will be G right here, and then AGC is right here. So we have AGC, and we know that AGC codes for serine. Yeah, this is what we have here, the serine. Now, one thing that you need to know is that those are the coding codons, or the sense codons. What it means is that these codons here, most of them, and there are 64 of them as a total, you can count them, but what we have here is that we have all those codons are involved with the ribosome interaction with the tRNA. These codons will interact with an anticodon found on the tRNA. And then we have all those amino acids here, and we have 20 amino acids. What you can tell is that there are, for some amino acids, there are more than one codon, or more than 4 even. Like look at leucine here, right? We have a total of 6 codons for this leucine. We have 1, 2, 3, 4, 5, and 6 here. But in some cases, we have only a few codons. So if you look at arginine, for example, arginine only has 2 codons here. So it has 6. But some of them have less than that. So for example, glutamic acid here does have 2 right here. Yes, only 2. Okay, so So what I mean to say here is that some codons are kind of special, and what they do is they don't code for necessarily an amino acid. So you see here, some of those codons here are stop codons. We have 1, 2, 3 right here. There are stop codons. So whenever the ribosome meets these codons as seen right here, right, when it meets these UAG, right, UAG right here, when it meets this, it stops. It doesn't add a new amino acid. It just stops there. And that is true for the other stop codons as well. So if it meets UAG, UAA, or UGA, then in that case, the ribosome will stop moving along on the mRNA. There is another very important and special codon as well, and that is the AUG right here. So this AUG codon codes for methionine, and we don't have an example here in our RNA here, but the start of any mRNA, so the first codon to be read by the ribosome, so the start codon right here, is always AUG. So that AUG is always coded. So every polypeptide starts, at least potentially starts, with a methionine. Sometimes a methionine is removed, but at least we know that AUG, as seen here, is always the start of any polypeptide. Okay, so what we have seen so far is that when we look at a piece of mRNA, as seen here, we know that this mRNA, in order to be transformed into a bunch of amino acids that will produce a polypeptide or a protein, it goes from one codon to the other. And in doing so, in order to add the right amino acid here that corresponds to this codon right here, the ribosome will read those three letters here, or those three nucleotides, and move on to the next, and then to the next, and then to the next. Now, the question here is this. It is possible, and because your DNA and my DNA and the DNA of all living organisms is usually super long, it is possible that sometimes the DNA will be changed a little bit, right? And because the DNA will be changed, it is possible that through the process of transcription, it is possible that the mRNA that will be produced from that changed DNA might have a little change here and there, right? So it is possible that sometimes some of those nucleotides here will be a little bit different than what we would normally see for most of the proteins. So these changes here are called mutations, okay? Mutations are not always a bad thing. Mutations help in creating diversity in nature. It actually is very important for evolution when you think of all the species of the animals and the adaptation to different, and plants as well. And when you look at different climates, the different living environment and everything that happened, and where we can find life, well, all of this is due to different mutations that have been selected throughout the ages, and then the most successful mutations led to more successful individuals, and those individuals, of course, survive these conditions. And all of that is due to different mutations. And all of our DNA at any given time, as potentially can be mutated, can be changed a little bit. And sometimes these mutations don't do anything, because mutations happen in regions where there are no genes. So that's one thing. But so those are called silent mutations, where nothing really happens. So basically, if there's a mutation, you would not even know. But in some cases, sometimes, and most of the time, if there is a change in the DNA that is a coding part of DNA, which leads to the formation of a protein through mRNA, so a coding gene, basically, that's what That's what we call that, genes. And then if we have a mutation in those genes, sometimes these mutations might lead to a very, very different and sometimes dire outcome for the cell. And this is what we're going to look at right now. So the next part of this video will deal with the different kinds of mutations that we can find in cells. Now, one thing that you really want to remember here, and that's very important for this part here, is that DNA leads to the formation of a polypeptide seen here through the formation or the transcription of something that we call mRNA. Okay, so that's important. But what we see here, of course, is the DNA basis, because this is where the mutations happen. Now, let's look at what happens here. Okay, so we're going to look at this part here is called a missense mutations. What it means, missense, is that it makes a mistake. It transforms one amino acid for another. Okay, so let's look at something very simple here. We have these sequences, DNA bases, that will lead to the formation of codons, and these codons will be read by the ribosome. We don't have this process here, but by now you should be familiar with that, and you should remember how and know how all of that happens. So what we have here is a bunch of sequences, CAT, CAT, CAT. Now, if you look into your genetic code, we know that CAT codes for an amino acid that is called histidine right here. So basically what we have here is a bunch of histidines one after the other. It just works for this example right here. So what we're going to see here is that when we have a change in DNA, and that change might happen for different reasons. Most of the time it's because in the process of copying of DNA replication, it's possible that the DNA polymerase 3 might make a mistake, and instead of adding an adenine right here, we'll actually add a cytosine. Or sometimes it is in the process of copying mRNA, so making mRNA from a DNA template. And in this case, the RNA polymerase would add, instead of adding a would add a C right here. And those mistakes can happen all the time. So what would happen in this case? Well, in this case here, if we replace the the base here, that's A, that you should call for histidine right here, if we change this, say, by another nucleotide here, that is C, well, this CCT here will lead to a very, very different amino acid. And that amino acid here is proline, right? So you see we have a bunch of histidines right here. This is what we should see here. But what we see now is because of only a single base change leads to the introduction, or the fact that one of these amino acids incorporated inside the growing chain of polypeptides is now going to be a proline. And in that case, of course, this is an incorrect amino acid, which of course may lead to malfunctioning protein. I say may here, and that's important, may, because it's not always the case. Sometimes it could also enhance this function of that protein. Basically, it makes it better. But most of the time, it will have some impact, and that impact, unfortunately, might be malfunctioning. And this is if this protein is found in a key cell, say brain cell, for example. What is possible that one cell might misfire and not do what it's supposed to do. If there's only one cell in your brain that does that, well, so be it. But when these cells start to reproduce and make 2 cells and then 4 and 8 and 16, etc., well, you can imagine now that in some cases that can lead to very difficult situations. Now, moving along the same scenario, what we're going to do here is look at something that's called a nonsense mutation. So basically, a nonsense mutation leads to the appearance at the end of the mRNA. It leads to the incorporation of a stop codon. So now remember what a stop codon is. A stop codon is when we have a ribosome. So this is my ribosome here, and then this is my mRNA, and this is my stop codon right here. So once the ribosome hits this stop codon here, this is where it stops. At this point, at the stop codon, the ribosome is going to deassociate from the small subunit. Those two subunits will not interact anymore, and the mRNA will be liberated from this, what was usually a complex, right? The three of them were interacting with one another, as we can see here. So when we have the stop codon right here, this stop codon leads to this. So it leads to the termination of translation or protein polypeptide growth. So what we have here in this case, we have an mRNA, and that mRNA, not the mRNA, but the DNA bases that we have here, those DNA bases, when transformed into an mRNA and then a protein, lead to the formation of a bunch of a sequence here. And for the example we have here, leads to glutamine, right? So glutamine one after the other. Okay, so this is not necessarily a functional protein. This is just as an example to show what could happen. So what we have here is that we have a change here, and that change happens in this base right here. And basically the C base, the cytosine base here, is now replaced by a thymine. And of course, this will lead, what we would normally have is CAG coding for glutamine here. But then what we have in our case here is a TAG, which of course, when translated into mRNA, leads to a stop codon. Remember, this here is DNA. And DNA here, remember that DNA is not what is being read by the ribosome, but the mRNA. So please look at this sequence here and make sure you understand why it is TAG. So, and then that leads to a stop codon. And of course, this stop codon here tells the ribosome to stop. So what we have here, we still have the CAGs that still need to be read, right? This is part of our growing polypeptide chain. These amino acids here might be very important for the function of a protein, but unfortunately, what we find here is that we have a stop codon right there. And the stop codon will eventually, for sure, lead to a shortening of the protein. Now, question for you, the answer I think is quite obvious, is, well, what would happen if we have a protein that is shortened, that is missing bits and pieces of it, and that sometimes it can be huge, long stretches that are missing? What do you think is going to happen, right? Of course, you know that the function of this protein can be hugely impacted, can lead to an absolutely non-functional protein, and not having that protein in one cell can be catastrophic if the cell needs to use that one gene for a function or another. Okay, now let's look at another type of mutation. In this case here, it is something that we call insertion mutation. And insertion mutations are different because they involve the displacement of all the amino acids after. And before I explain this, I need to talk to you about a concept that is called the reading frame, right? So the reading frame. So the reading frame is what the ribosome goes through. So when we look at the reading frame here, the ribosome on the mRNA, and again, please remember this is DNA, right? So it's just because mutations happen in DNA, but remember that in between here, we have mRNA, right? So that's very important. So the reading frame is this. The ribosome on the mRNA, what it's going to do is going to look at this codon here, the first one, and then it's going to move on to the next one, and then to the next one, and the next one, right? So basically, it goes from one to the other. It reads one, it reads the next one, it reads the next one, and reads the next one. This leads to the incorporation in the growing polypeptide chain of the incorporation of histidine one after the other, because the reading frame seen right here, the reading frame says you read this one, and then you read this one, and then you read this one after, and so on and so forth. So that is what we call the reading frame. Now let's look at this very interesting mutation here. So what we're going to do here is right in between the T and the C right here. So right in between those two nucleotides here, the T and the C right there, what we do is we inserted a adenine right there. Okay, so what happens to our reading frame in this case here? Remember, look at that reading frame here, 3 3, and then our normal reading frame would have been this here, and then this one right there after. However, we added a new base there, so the new reading frame reads like this: ACA. Remember that a ribosome reads 3 by 3. So it will read ACA, and then the next reading frame after that, the next 3 ones will be this one, then will be this one, and then this one. So what we have done is we have shifted to the right. We have shifted to the right the reading frame. And this, of course, will lead to the incorporation of an incorrect amino acid sequence. It leads to the incorporation of the threonine here, and the cell might not have anything to do with that. What I mean by that, to do with it, what I mean by that is that this new mRNA here and then this new protein that we see right here might not have a function, because what we need is not a threonine that we need here. We need a histidine, and we need histidines all after. You see, this single shift by adding only one nucleotide here, only one adenine here, created one amino acid that's different, threonine, that is different from histidine, of course, and then a bunch of serines after that. Of course, this changes the whole sequence, and then when it comes down to proteins, when you change the sequence of the proteins, of all the amino acids, this has a huge impact on the function, because the form is now changed, the function will be changed as well. This can lead to a non-functional protein or misfunctioning, and in some very, very, very rare cases can lead to an improved protein, but that's not very often. Okay, so most of the time this will lead to a problem for the cell. So what we have here is another kind of mutation that still leads to a change in the reading frame. So remember what the reading frame is. The reading frame is this. We have this codon here, and they're read one after the other, leading to the incorporation of all histidines here in our protein. Now, in the previous one, we saw that there was an insertion. So and we shifted the reading to shifted the reading of the ribosome, the traveling of the ribosome on the mRNA. Well, we're going to do the same thing here, but in this case here, it's by deleting, it's by removing an a nucleotide. Okay, so let's look at this. What happens here? Well, what we have here is a CAT sequence right here, right? We have the CAT sequence here that should lead to the incorporation in the polypeptide chain of a histidine right there. But what we're doing here in this case is that we are removing the A that should be right here, that A right there, we remove that. And of course now the new codon in the mRNA is not CAT anymore that leads to histidine, but it is CTC that we have here that leads to leucine. And then after that, all of the other codons after this will lead to the incorporation of isoleucine inside the growing polypeptide chain. So for the exact same reason that we have seen in the incorporation of a base, a shift by incorporation, this will lead to incorrect amino acid sequence, which may produce a malfunctioning protein for the exact same reason, but because we are changing the reading frame, but in this case, it's because we are deleting an amino. Now, to continue with the same subject of reading frame amino, not amino acids, but reading frame problems on the mRNA that leads to the incorporation of faulty amino acids, the wrong amino acids, we have here a change in our mRNA, not because we are inserting anything or not because we're deleting anything, only because what happens here, we have a shift in the reading frame. So what happens is that our ribosome might, for a reason or another, skip the first base here, which should lead normally to the incorporation of histidine, the CAT here, right? What it does is, instead of reading CAT as we see here, what it does is that it starts to read at ATT right there, right? So ATT leads to the incorporation of an isoleucine here. So it is a frame shift mutation. The reading frame has shifted to the right in this case. And of course, this will lead to very different types of amino acids. And then you see that with the sequence here, and we can tell, for one thing, at the beginning, is that compared to the other mRNAs we've seen before, that always led to the incorporation of the same amino acid, to use as an easy example for everybody to understand. What we have here in this case here, we have our mRNA, and then our mRNA, when it's read, it transforms into a protein that has different kinds of amino acids incorporated, right? Well, of course, this sequence of amino acid here is very, very important. This will allow the protein to have a specific function. But when you shift these mutations, either by deleting, by inserting, or because the ribosome doesn't start where it should be, shifting for one reason or another, then what happens here is you see that all of those amino acids here are completely different from what they should be. And of course, this will lead to a very, very different protein. That protein might have a function, most of the time it won't, but it could have a very different function, and that function could be detrimental to the cell. Okay, so that is important. Now let's shift gears a little bit and talk about other kind of mutations that can happen. So in the previous few slides, we looked at differences at the nucleotide level in the DNA. The coding sequence led to a faulty or potentially faulty creation of a polypeptide chain that would be probably in most cases non-functional. There are other types of mutations that can happen, and not all mutations are created by the fact that some bases are changed in some way or because they're deleted or inserted. Okay, so there are different other kind of mutations, and this is one of the other kind I'm talking about. So what we have here in our example right here, we have a DNA molecule, and then of course we recognize all those 4 letters here, and then we see that that these are 2 strands of DNA, 3', 5', 5', 3', etc. And what we have here, as seen many times in all DNA sequences, we have 2 thymine that are connected to one another, right? So in our sequence, we have T, G, TC, G, GC, and then T, and T as seen right here. So these 2 nucleotides, what can happen is that when these 2 nucleotides, when excited by UV light, so light from the sun or light that has enough energy in the UV spectrum, we know that light has a certain amount of energy to it. And then when these 2 nucleotides here, when we have those 2 thymine and thymine right here, and another thymine right here, we know that UV light can create links between those 2 thymines here, right? So it will chemically it will create a covalent bond between those two thymines. So the thymine number 1 here and thymine number 2 there. There is a covalent bond that can be seen right here in the middle. And of course, when this happens, normally what we would have is those two nucleotides here would be, like this, not connected, not doing anything here. But what happens though is that when these two nucleotides are connected with one another, then they do interact as seen here. And of course, what happens in this case, look here, it really deforms the DNA molecule. It really deforms it. And what do you think is going to happen when these two nucleotides here are connected with one another as T and T, connected covalently, deforming that DNA molecule? What do you think is going to happen when it's time to replicate DNA, or when it's time to create an mRNA from that DNA? Well, you can imagine that it's going to be a problem, right? Well, what can happen here is that this can lead to the fact that the enzymes that are implied, that are very important for DNA replication or mRNA formation, the RNA polymerase, might not read this. And then we might be missing some very important information. The cell might lose some very important information there. And that, of course, can be very, very highly problematic for the cell. But fortunately for us, most of the time, we will have an enzyme that will be, will recognize these thymine dimers with our, look here, we have our deformed DNA right here because of the thymine dimers here. What the photolyase will do, it will recognize these sequences, it will recognize this form right here that is now deformed, will interact with it, and then break those bonds that are creating between the thymine dimers, and then return the DNA to its normal shape right here, so that the cell can use the information that is encoded in that DNA, and of course create proteins that are necessary for the cell function. However, it happens that sometimes these functions, those enzymes here, stop to function. Okay, they stop to function because let's say that there is a mutation in that protein here caused by either deletion, insertion, frame shift mutation, etc., nonsense mutations, etc. It is possible that a cell might completely lose this enzyme here that would now be not functional. Now, what we're going to look at right now is the consequences of losing very important proteins like that. So let's look at that. What we have here is a single cell, right? And that single cell right here will normally go to the cell cycle, right? So it will go from G1 to S to G2 and then to mitosis, and then back again to G1. Okay, so we will go from one cell that would lead to two new daughter cells that usually should be healthy. So cells should proliferate. But when we have a mutation, this mutation might, instead of having some stops in the cell cycle, remember that the cell cycle is highly regulated and it has some very important checkpoints where the cell cycle will stop at some points and stop the cells from proliferating. So let's say that the checkpoints are mutated, and those mutated checkpoints, do not stop the cells when they should be. Well, what will happen to those cells? Those cells will proliferate. Those cells will grow, and then from 2 cells we'll have 4, and then we'll have 8, etc., etc. So we will start to have a little mass of cells there. And then mutation after mutation after mutation can happen. So once you have a mutation that allows the cells to grow without being controlled, that probably will lead to the formation of new cells that will not have the control as well. And then mutations might accumulate over generation and generations of cells. And at the end, as you go from one mutation to another, that can lead to cancer. That can lead to cancer cell. And of course, cancer is something that everybody's afraid of. And of course, that's because, for a good reason, that means that cells are now growing inside a body and being totally unregulated. There's no regulation whatsoever. The cells grow when they should stop growing, and then they will do damage to tissues. They will compress tissues. They will move from one tissue to another. For example, they might go from a liver cell to brain to go through the barriers and get in your brain, etc. Or bones. So that's important here. Now, one thing that you need to know is that a single mutation is very unlikely to cause cancer. Like just one single mutation will not lead to cancer, but that single mutation might lead to a cell that is not checking as much as it should, that will lead to more mutations, and those mutations will accumulate one after the other. The result of all those mutations happening one after the other, after generation and generation and generations of cells, will and can lead, can lead to cancer if your immune system is not able to recognize those cells and get rid of them. So fortunately for us, we have genes that are important for the cell to remain healthy. So evolution has selected genes that will protect the cells from transforming into cancer cells. That's called cell transformation when it gets into a cancer form. So when the cell starts to accumulate DNA damage, as seen here, DNA damage, or other types of cell cycle abnormalities. So remember that the cell cycle is highly regulated when cells go from G1 to S, and then they go to G2, and then eventually go to mitosis, all the phases of mitosis, right? Remember that there are many checkpoints at different spots where the cell will check and make sure that the DNA is actually functional and there's no damage there, right? So there's no damage. Well, that protein that is responsible for this, for checking DNA damage, is called p53. And that's why p53 is called a guardian of the genome. So what it will do is that when it detects that there is DNA damage, it will stop the cell cycle. It will stop the cell cycle and then will make sure that DNA gets repaired. So a bunch of enzymes are going to interact with DNA, including lyase, as we have seen before, and DNA will be repaired, and then the cell cycle can restart from where it was before, right? So in that case, we repair the damage, and then we ensure that there is genetic and cellular integrity and stability. That can lead, so to make sure that the first cell that led to those two cells that will lead to two cells after that, we know that this cell here is now healthy, and that the genes and the DNA found in that cell is not damaged, so that the two daughter cells found here will have the same genetic material and the same integrity as the first cell in the beginning. However, in some cases, the DNA is so extensively damaged that the cell has to make a decision, right? It's like, it's like a car crash in a way. And when, if you crash your car, well, you might be able to repair the car, and it's going to cost you a certain amount of money. But it doesn't take much for the amount of money that the repairs will need to be more a higher value than your car is worth. So we say in that case that it's total loss, right? You can't repair a car like that because basically buying a new car is less expensive, and no insurance company will go that way if you are insured, hopefully. So so it happens at the cell level as well. It is possible that in some cases the DNA damage is so extensive that the cell will decide to go into apoptosis. So basically we'll call it a total loss, and we eliminate the cells that are damaged. So if there are too many mutations, then that cell will go into apoptosis and will be eliminated from the body. So finally, in a very quick nutshell, this is the apoptosis process. And please note that the apoptosis process is a highly complex process. It involves a lot of different proteins. It's highly regulated. It's not an easy process and involves a lot of different partners. What I want to show here, though, is the basic steps that lead to apoptosis. And apoptosis basically is at some point. So this is our normal cell here. So as we can see here, this is our normal cell with the nucleus, the mitochondria, etc. What's going to happen is that in some cases where there is a lot of damage inside that DNA right here, and then P53 decide, you know what, I don't think I'm going to go repairing. I think instead of repairing, we're just going to call it quits here and then kill that cell, right? So the cell will eliminate itself. So there's a genetic program that will be activated. So the cell actually decides to do that and commits something that we're calling cell suicide. So basically what's going to happen is that the DNA inside the nucleus here will condense. It's called pycnosis. And all of these, of that cell here will now shrink and form smaller pieces here that are called blebs. And then this is now part of our one cell. It's broken into smaller pieces. And all of those smaller pieces here will now be digested by some of your cells here that are called phagocytic cells. And so those cells here will now literally eat and digest what was part of a cell before. Now, remember this, the apoptosis is an important process involved in homeostasis, so the maintenance of your body functions. It happens when DNA is extensively damaged and cannot be repaired. It is a genetic program that will lead to the destruction of your cells from normal cells to parts and bits of pieces of this cell that will now be digested by phagocytic cells, okay? And those cells here, the phagocytes, will digest that, and that abnormal cell here, that could have led to cancer. It could have led to cancer, will be eliminated. So at any given time in your body, you do have cells that are right on the verge of turning cancer, but it doesn't mean you are going to get, quote unquote, get cancer. You don't, because we have the right mechanisms in place to protect ourselves from these cells. Okay, so we are protected from these cells by very efficient mechanisms. So that's why cancer is not as frequent as it could be.